Hi, I'm Mr. Williamson. This video is about the Krebs cycle, aerobic respiration. Before we get started, make sure you take good notes. There's a lot of information in this and a lot to digest, but don't worry, I got you covered. I would suggest Cornell-style notes. I found they work best for students. Make sure you rewind or pause the video if you don't understand something. Ask good questions in both Edpuzzle or in the classroom. Let's get started. Before we go any farther, quick review. The last video we talked about glycolysis. Glycolysis takes place inside the cytoplasm. It's not in the mitochondria yet, but inside the cytoplasm. It starts out with glucose, which we get from food. We use the energy from two ATP molecules. Start with glucose. We use the energy from two ATP molecules to split it into two 3-carbon molecules, pGal. After that happens, we split off two hydrogens, which we store in NAD. Don't worry about what NAD is. I'll explain it later, but they become NADH2. In the process, we actually get enough energy to make 4 ATP, so now we're up 2 ATPs, so we've actually made a profit here, and as a result, we end up with pyruvic acid. And this is what we're going to be talking about, is these two pyruvic acids. And actually, we're only going to follow one of them. Okay, we're going to take a look at one pyruvic acid. What happens to that one pyruvic acid when it goes in through aerobic respiration? So the first thing that happens is the pyruvic acid moves to the mitochondria, to the inner membrane of the mitochondria. Remember the mitochondria? Powerhouse of the cell, all that stuff? Well, this is why it earned its name. Okay? So it goes in the intermembrane, in the membrane that is right in here, the inner membrane. Not the outer membrane, not the outside, but the inner membrane, where all the little nooks and crannies are that you always draw. What is this thing called? Mitochondria. Couldn't remember the name. Why do we always draw mitochondria like this with the inner membranes? Because it has them and they're an important part of the ATP production. So we're taking that pyruvic acid that we got from glycolysis, from breaking down a glucose into two 3-carbon molecules called pyruvic acid, storing the hydrogens on NAD, and we get the two ATPs. We're going to move that pyruvic acid into the inner membrane, this part of the membrane of the mitochondria. The first step of aerobic respiration is to change pyruvic acid into acetyl-CoA, and a carbon dioxide molecule is removed from a 3-carbon. Again, I have to count. Let me take a moment here. In the middle, I'm interrupting right here. That's where I'm stopping us. I have to account for every atom in glucose, C6H12O6. I have to explain where it goes. From the time the cell enters the cell, every C, every H, every O, all 6 C's, all 12 H's, all 6 O's. I have to tell you where they go. Where do they go? Okay? And I'm going to do that. So, first thing, we're going to change it into a thing called acetyl-CoA. Don't panic. Don't panic. Okay? And we do that by breaking off a carbon dioxide, all right, from a 3-carbon. So it becomes, if you take a 3-carbon, you break off 1 carbon, you get a 2-carbon. It's math, all right? And that's what acetyl-CoA is. It's a 2-carbon molecule. We also remove 2 more hydrogen ions and add them onto NAD. Now, you could write all that stuff down. Don't worry about it. Besides, picture's worth 1,000 words. So we have here pyruvic acid outside in the cytoplasm. All right, and let's pretend right now that this line is the cytoplasm, and inside that, on the bottom below that, is the mitochondria. So this is the mitochondria. So it starts with pyruvic acid. It goes inside the mitochondria, into the inner membrane of that mitochondria. When that happens, we break off a carbon dioxide. Where does the carbon dioxide go? Well, I asked you what you eat, why you eat, and you said, or breathe, and you said, well, if we don't, we die. Okay, well, now you have explained to you if you eat because you need glucose. Another question for you. Why do you exhale carbon dioxide? A lot of carbon dioxide. You guys know that. Breathe in oxygen, breathe out carbon dioxide. And I assume that you probably think that the oxygen is turned into carbon dioxide. It's not. We haven't talked about oxygen. Why do you exhale carbon dioxide? It's right there, guys. We're breaking it off of glucose. Carbon dioxide you exhale comes from the glucose that you're breaking down to get energy. We broke it down to pyruvic acid in the cytoplasm, and then we move it into the mitochondria, the inner membrane of the mitochondria. We break off a carbon dioxide, exhale it, okay, and we break off 2 hydrogens. Where do they go? They go NAD, which becomes NADH2. Where does that go? Don't worry about it. We'll get to that. Don't worry. Okay? Just need to know that we've lost 2 hydrogens, carbon dioxide. What does it become? It becomes this acetyl-CoA thing. Do you need to know that? No, you don't need to know that. No, you don't need to know any of this. No, I'm just explaining to you. Don't worry about it. Don't look at this and go, oh my God, I'll never get this. Put your pencils down, guys. Put your pencils down. Okay, you might want to draw this, but you don't have to know it. You need to understand. I'm going to add to this diagram. So, yeah, you might want to draw this, but make it simple. Don't worry about it. I promise you. All right? So again, to summarize this, start with pyruvic acid, which we got from breaking down glucose and glycolysis in the cytoplasm. Pyruvic acid moves into the mitochondria, into the inner membrane. We break off a carbon dioxide, all right? So it becomes a 3-carbon, becomes a 2-carbon. You exhale the carbon dioxide, carbon dioxide is gone, and we break off 2 hydrogens. Where do the hydrogens go? NAD. We're going to store them for later. All right? So that acetyl-CoA now enters a biochemical pathway called the Krebs cycle. Sometimes it's called the citric acid cycle. So if you see Krebs cycle or if you see the term citric acid cycle, it's the same thing. All right? It's named after this guy, Sir Hans Krebs. He died in 1981, so it's kind of recent. I mean, for you guys, it's like, whoa, but I was like, not dead. I was born. And he named it after himself, and he's going to cause you all kinds of problems if you continue in biology because you're going to need to know Krebs cycle. You don't need to know Krebs cycle, but he's dead. All right? So that 2-carbon acetyl-CoA enters the cycle, and okay, look, you don't need to write this down. Don't write this down. If you want to, by all means, if that floats your boat, do it. Don't do it. We're going to combine acetyl-CoA, which we got by breaking carbon dioxide off of pyruvic acid, and we're going to combine it with a 4-carbon thing called whatever. I don't know. I don't care. Okay? And it becomes a 6-carbon called citric acid. All right? Picture, 1,000 words. Here you go. We started out with acetyl-CoA. Start with adding a 4-carbon. It becomes citric acid, a 6-carbon. How? 4 plus 2 equals 6. All right? 4 carbons plus 2 carbons equals a 6-carbon. Again, I have to be able to explain to you where every atom goes and where it comes from. So right about now, you should be going, yo, Williamson, what the hell, where did that come from? I'll answer that later, but I will answer it, okay? Got to explain where it came from, and I will, but not right this moment. Just understand that, and you don't need to know the names of these, understand that we took a 4-carbon, we added it to a 2-carbon, and we ended up with a 6-carbon. We're in the Krebs cycle now, the citric acid cycle. Don't worry about knowing this. You will not need to go walk me through. You won't need to do that. Okay, so the Krebs cycle, carbon dioxide, another carbon dioxide is removed from the citric acid. If you remove a carbon from a 6-carbon, you get a 5-carbon called ketoglutarate, blah, 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 blah. Okay, and then at the same time, 2 more hydrogen atoms are removed. Where do you think we could store hydrogen ions? Oh, I don't know. How about NAD? So, yeah, again, blah, 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 all this, blah, 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 blah. Picture, 1,000 words. Here we go. So we started out with A citric acid, a 6-carbon. All right. And we go along and we take off carbon dioxide. You exhale carbon dioxide. Where'd it come from? From glucose, breaking it down, getting energy to make ATP. That's where it came from. All right. And then we break up 2 more hydrogens. Where could we put those? Oh, I have an idea. How about we put them on NAD, which becomes NADH2. And it's probably on top of my head or behind it. Who cares? I'm probably like, it's right behind me, I bet. Am I right? Yeah. Anyways. Okay. So if we remove a carbon dioxide from a 6-carbon, what does it become? It becomes a 5-carbon called Glycolysis something. Who cares? I don't care if you know the name. You're not supposed to know the name. Get the idea of the process here. You with me? This would be a real good time for me to put a question in Ed Puzzle. I wonder if I'll do that. I don't know. Okay, so we take that 5-carbon and we remove another carbon dioxide. Oh, I don't know. Let's see. If we take a 5-carbon and we remove another carbon dioxide, which you exhale, okay, it becomes a 4-carbon. 5 minus 1 is 4. Okay, it's called succinylate acid or something. I don't care if you know the name. Not important. Okay, and at this point, we actually got enough energy to make 1 more ATP. So we made 2 we got a profit of 2 in the glycolysis, and now we're getting 1 more. Now we're up to 3. We are rolling in ATP. No, we're not. We need more. Okay, picture's 1,000 words. Start with the 5-carbon, okay, and break off 2 hydrogens. Where could we put these 2 hydrogens? I don't know. How about put them onto NAD, which will become NADH2. What a shock. Oh my God, whenever we get rid of hydrogen, we put it on NAD. Works for me. Okay, so plus the carbon dioxide, where's that go? You exhale it. It becomes another 4, it becomes a 4-carbon molecule. So we've gone, and we're just, again, simple math. Start with a 5-carbon, subtract 1 carbon, becomes a 4-carbon. I know. It's not that difficult. You don't need to know this. Just kind of understand it. Okay? Plus, we get enough energy to make ATP. We turn ADP. Oh, I can do this. That's good, Williamson. Good spelling there. ADP. We get enough energy to make it into to attach a phosphate to ADP, diphosphate, and turn it into ATP, adenosine triphosphate. We now have 1 more ATP molecule. So 2 more hydrogen ions and blah, 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 blah. 4-carbon called malic acid. That's what this 4-carbon is going to be called. Okay? Now, how's that look? Looks like this. Right here, we have a 4-carbon here, and we roll into take out 2 hydrogens. Where could we keep those? Yeah, you said NAD, didn't you? No. See, if you were paying attention to the previous slide, we started on a thing called FAD. Why? I don't know. I figure it's just some way of making high school biology students' lives miserable, but we don't store in an FAD, NAD, we store on FAD. Okay? Which becomes FAD to H2. Okay? Don't worry about it. Don't worry about it. But you might want to kind of think, just kind of think, that this goes to NAD, and this one goes to NAD, and this one goes to NAD, this one goes to FAD. That's it. You know, do you need to know it? It's not going to be on a test. No. Do you need to understand it? It's probably a good idea. Try to get the concept, okay? And we end up, because we haven't removed a carbon, no carbon dioxide exhaled, still have a 4-carbon. Can't subtract something. If you don't subtract it, it stays the same. All right, so the next thing, 2 more hydrogen ions are removed, and we end up with this oxalic acid, which should sound a little familiar, because I talked about that earlier, okay? So again, picture worth 1,000 words. We remove 2 hydrogens. Where would we put 2 hydrogens? Well, I don't know. NAD. What an idea. Put this one in NAD, Oh, that's terrible. I can't believe that's not an N. Let's try that again. Sorry about that, guys. NAD, which becomes NADH2. And just to help you out, this is stored on NAD. This one is stored on NAD. This one is stored on NAD. And this one, for some reason, is stored on FAD. Why? I don't know. I don't care. I don't care. This one, NAD also. Plus, we haven't removed a carbon. We did not exhale carbon dioxide, so it stays the 4 carbon. What do we do with that? Well, the oxalic acid is the molecule that joined acetyl-CoA in the form of citric acid. Remember I said where I have to tell you where I got that carbon to make this 2 carbon and that 4 carbon into a 6 carbon? It's a cycle. I get the 4 carbon, the 2 carbon, put them together, you get 6 carbon. And the next pyruvic acid comes in and starts the cycle over again. That's it. That's the Krebs cycle. Okay? You don't need to know all the malic acids and the acetyl-CoAs and the citric and whatever. That's all words. I don't care. Understand the principle. So that would complete 1 cycle. But remember, we had 2 pyruvic acids per glucose. C6H12O6, we broke it into 2 3-carbon molecules. So when one goes through, another one has to go through, so you get the whole glucose molecule. Because each glucose molecule requires 2 cycles of that Krebs cycle so that we account for it, okay? So, quick review. Each pyruvic acid goes through the cycle once. Each pyruvic acid, not each glucose molecule. Each pyruvic acid goes through once. But there are 2 pyruvic acids per glucose. So they go through twice. Okay? Hydrogens are stored, broken off, and they're stored someplace. And this is actually what we're doing here. We're trying to store them on NAD and one of them on an FAD per pyruvic acid. So they would be like 2 FADs and a whole bunch of NADs. Plus, we're exhaling carbon dioxide because we're breaking off carbons, and you exhale that carbon dioxide every time you breathe out. It's a waste product of metabolism. So the question is, what happens to the NADs and FADs? I've explained to you where the carbons and the carbons go, and I've explained to you, you exhale them. But what happens to those NAD FADs? And that is actually the whole purpose of the Krebs cycle, is to get those NADs and the FADs. We need to talk about those. Well, they're going to travel to a thing called the electron transport chain, which is located in the outer membrane of the mitochondria. And that's the subject for the next video. So let's take a quick review. We talked about the products of glycolysis, the pyruvic acid. We also got an additional profit of 2 ATPs, and we got some NADH2 stored. And then that pyruvic acid goes into the mitochondria to the inner membrane, where it goes through the Krebs cycle. And we break off a carbon dioxide, and it becomes a 2-carbon molecule. But we attach it to a 4-carbon molecule. So now we're back to a 6-carbon molecule. We exhale some more carbon dioxide. We go to a 4-carbon molecule. And along the line, we're breaking off hydrogens here and there. And we're storing them on FAD. And then we break off, we're a 5-carbon, we break off one more carbon dioxide, which you exhale. And we break off some more hydrogen. And then we break off another hydrogen. But this one we're not putting on an FAD, on NAD. We're putting on an FAD. Why? No clue. Don't care. Break off another hydrogen stored on NAD. That brings us to a 4-carbon molecule, which we combine with the next pyruvic acid that comes in, making a 6-carbon molecule. That's the Krebs cycle. Yes, it seems like there's a lot there. There's a lot of words there. Blah, 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 blah. Who cares? You probably want to go back and maybe copy down that diagram. That would be a good idea because it would help you remember things. Okay? Important is the NADs and the FADs. Plus those, we got an extra ATP out of it for every pyruvic acid. So we got 2 ATPs in glycolysis, and then we got 1 pyruvic acid ATP, and then we got another pyruvic acid ATP. That's a total of 4. We have made a profit of 4 ATP molecules from 1 glucose. Like I said, we are rolling in ATP. No, we're not. Not even close. We're not done yet. Where do the NAD and the FADs go? The electron transport chain. We're going to talk about that later. Hopefully you took good notes. Make sure you ask good questions at the end. If you have any, put them in. I want to answer them. Or ask them in class. Again, hope you took good notes. Good luck.